Pretty much every day, I fantasize about walking away from my computer and home office and heading out into the wilderness for a better life. I mean, I spend more time in nature than most folks, but I am still very open to moving into the woods, building one of those in-ground shelters that they show being built on YouTube and just calling it home. Since there are so many pragmatic obstacles with this option, though, I find that I usually just load up with nature hobbies and forest and tide pool days. Today's episode is funny because by the end of it, I am very clearly ready to devote myself to becoming a mushroom explorer, set up a genetics lab in my home, and begin to start discovering and describing mushrooms. It will be even more ironic if we look back at this episode in a year and I'm actually doing that. Today's episode is a lot of fun if you love mushrooms, especially psychedelic ones. Today's episode is equal parts storytelling and cutting-edge science. Alan is kind and generous, and the episode is a lot of fun. You'll hear that I clearly had a blast, and I hope you do too. If you want to learn more about cannabis health, cultivation, and technique efficiently and with good cheer, I encourage you to subscribe to our newsletter. We'll send you new podcast episodes as they come out, delivered right to your inbox, along with commentary on a couple of the most important news items from the week and videos too. Don't rely on social media to let you know when a new episode is published. Sign up for the updates to make sure you don't miss an episode. As the cannabis market continues to compress and crumble over much of the United States, there are fewer and fewer cannabis and mushroom companies that have a budget for marketing. If you know of a cannabis or mushroom company not run by cops, rats, or chemical fertilizers, who should be an advertiser on Shaping Fire, please let them know and let me know too. It is by working together that regenerative cultivation expands. And we're now offering a smaller entry package of commercials, so we're able to include folks with smaller budgets. Thanks. If you're here for the first time because of the mushroom topic, welcome. We do quite a few mushroom episodes here at Shaping Fire. Be sure to check out Shaping Fire episode 85, Psilocybin Mushrooms and Brain Chemistry with Dr. Miyabi Shields. Episode 76, Comparing Psilocybin Microdosing Protocols with guest Adam Bramlage. Episode 22, Treating Brain Injury with Cannabis and Mushrooms with guest Dr. Ethan Russo. Episode 67, also with Dr. Ethan Russo, Migraine Therapy using cannabis and mushrooms. Episode 95, Psilocybin Mushroom Analytical Testing with guest George Selhorn. And number 70, Cannabis, Mushrooms, and Terpenes for Optimum Health with guests Dr. Ethan Russo, Tony Vazura, Jeff Chilton, and Berner. You are listening to Shaping Fire, and I am your host, Shango Los. We are in our 10th year of Shaping Fire, and this is episode 128. My guest today is Alan Rockefeller. Alan Rockefeller is a mycologist, photographer, and one of the most prolific citizen scientists working in fungi today. He's spent over 2 decades in the field, documenting, collecting, and helping identify thousands upon thousands of mushroom species globally. He's the kind of person who doesn't just look at mushrooms, he tracks them across ecosystems, photographs them in ways that'll make you fall in love with them, and then takes them back to his microscope and then right down to their DNA. Alan has described new species and has co-authored scientific work on multiple psilocybin-containing mushrooms. But what really sets him apart is how much of his work is right in the open. He's identified hundreds of thousands of fungi observations online, contributing to platforms like iNaturalist and helping everyday people turn curiosity into real science. He's also a teacher at heart, leading forays, teaching microscopy and photography, and helping people not just to learn mushrooms, but to see them differently. He gets people fired up about mushrooms. Alan has appeared in major media, including the fantastic Fungi documentary and the first season of Hamilton's Pharmacopoeia, too. He's been featured in outlets like National Geographic and the New York Times and so many other places. During the first set, we talk in depth about Psilocybe cubensis as a species complex, the split of the Psilocybe genus in modern taxonomy, and the factors that have led to nearly all modern cultivated psychedelic mushrooms actually being the same mushroom, just expressing itself in myriad ways. In the second set, we talk about evolutionary pressures on mushrooms, how new traits evolve, and why nearly all the current mushroom identification guides are offering information built upon insufficient descriptions. And we finish the episode with a discussion of wild versus cultivated mushrooms, the importance and variety of secondary metabolites in mushrooms, wood lovers' paralysis, mycelium versus fruiting body supplements, and the sheer joy that comes from mushroom foraging with ultraviolet flashlights. All today on Shaping Fire, episode 128. Welcome to Shaping Fire, Alan.
Thanks for having me on.
Yeah, so glad you could make it. I've been watching your Instagram the last few weeks, and you have been all over the place on the planet recently, and I'm glad to have caught you back home in the Bay Area. Let's get started right into the heart of things. You know, most everybody who is, at least in my audience, has come to mushrooms because of psilocybin mushrooms, why we're here today. And, you know, many of us find our way to loving mushrooms because of our joy found in the hallucinogenic aspects. And that certainly was for me. And as I started to learn all about the fancy and varied mushroom varieties and, you know, seeing what was, you know, going on down at the Psilocybin Cup in Oakland and seeing the different varieties, I was really shocked to find that nearly all of the different varieties that are being modern cultivated by the people I know are all actually Psilocybin cubensis variety. And that's shocking because they all express themselves in such different ways. How is it that the old school, like Golden Teachers and Be Positive and the modern Title and Penis Envy, and then even like the wild looking Enigma and others, they're all essentially the same genetic mushroom, even though they look so different? They're all cubensis.
Yeah, cubensis is most of what people grow, although there's a couple hundred species and people could grow almost any of them. But, you know, it's kind of like how Brassica olyrica is broccoli, kale, cauliflower, cabbage, and a dozen other vegetables. So it's just different cultivars of the same species.
And so for people who aren't as experienced with understanding how that happens, this is mostly through, selective breeding, the ones that are bred that you like, mutations that you take advantage of, and the growing environment and things like that, right? So these are intentional things that the breeder is doing to push and pull the genetics in different ways, right?
Yeah, just like people do with plants, but you can also do something that's a little bit more like saving pollen in your freezer, where you cross different strains. And so it's pretty easy to take two completely different cubensis strains, get them to mate on agar, and then have a new strain.
Mm-hmm. People try to cultivate their mushrooms in an environment that is most beneficial to the mushroom, because most of us, we're just looking for healthy and volume, right? If we were to grow mushrooms in a less apt environment and stress them or push them this way or another so they express different parts of their genetics, might we use that, using the ideas of nature versus nurture, using this idea that we're nurturing it poorly into this different form, might we be able to then capture those attributes in a new mushroom, or does growing it in a certain direction not really help with the developing which parts of the genetics are expressed?
Yeah, so when different environments will cause it to express different phenotypes, but it won't really change the mushroom at all. It'll just look a little bit different. So there are people like Julian Matucci who's doing experiments with gamma radiation, and he found out that the mycelium is growing towards the gamma radiation source, and that'll probably cause mutations and do all sorts of crazy things. But that's pretty rare. In general, people just grow them. But I've been trying to talk people out of growing Psilocybe cubensis because it's kind of boring. It's not really native here. It's not super potent, and it kind of hurts your stomach a lot more than some of the other species.
What are some of the other species that you encourage people to look into? I mean, there's hundreds of Psilocybe species alone, right? And somehow we landed on cubensis. So I guess this is a two-part question. Do you know why we landed on cubensis as the one we wanted to work with the most? And what are these other varieties that you encourage people to look at?
So cubensis is super easy to grow. It's really fast. So to grow it is kind of like maybe a 2-month cycle. And it also has, it's very heavy. So it has a lot of biomass. So if you're growing it for sale, it's really hard to make economic sense of growing anything other than cubensis. But if you're growing it just to take them or for medicine or something like that, I think there's a lot of other better choices. And they're not as big and chunky as cubensis, and they don't grow quite as fast. Mm-hmm. But you have about 100 different species in psilocybe. And then you have other genera like Paniolus, Gymnopilus, Pluteus, Foliotina, which is now Carnicibula, but there's all sorts of different ones that in theory could be grown. The best ones are typically in Paniolus or psilocybe, and Paniolus is actually really distantly related to psilocybe, so that is likely to be pretty different. And then also in psilocybe itself, you have, it's kind of split down, the genus is split down the middle genetically, and we call it clade 1 and clade 2, and they have completely different psilocybin gene clusters. So it's very likely that something that's from the other clade is going to be much more different than something from the same clade. But, there's a lot of different chemicals in these mushrooms that cause the effect. You have like a dozen different tryptamines and all sorts of beta carbolines and things. And we're just starting to figure out, what effect those chemicals have.
For people who are new to researching mushrooms, but they're excited about it and looking for something, we mentioned that there are so many psilocybe varieties to look at. What are a few that you think are engaging and show some promise for people who kind of want a leg up on where they should look instead of just kind of diving into the big pool of psilocybe generally? Like, for the people who are taking notes and are like, oh, I'm going to look at these because there might be some interesting stuff there.
Yeah, I think my favorite one is psilocybe zapaticorum. And so zapaticorum is described from Oaxaca, and this is one of the landslide mushrooms. And so in Mexico, you find it growing at the base of landslides. So kind of a really crazy, dramatic habitat. But they can be huge. They can be up to a foot tall, and they're extremely potent with about 3% psilocybin. So it is the strongest wild psilocybin mushroom that has been tested. And they're also really pretty. The stem is kind of shaggy, and the blue staining is very intense cobalt blue. And so these, I think, are much better for people to cultivate than cubensis if they want something that's better to grow. And they're not super hard to grow. They're about a 5-month cycle instead of a 2-month cycle, so they take a little longer, and they need a little bit higher humidity. Cubensis is kind of tolerant of it drying out a little bit, but other than that, it's pretty much the same. So I've been trying to talk people into growing something like psilocybe zapaticorum, or psilocybe subtropicalis is another one that's closely related to zapaticorum, also found in southeastern Mexico. And in cultivation, that one's been measured at up to 5% psilocybin. So that one is about as strong as zapaticorum, and it's smaller. People have been growing it for quite a while, and that's probably about as good as far as the experience goes. But, I noticed those species don't hurt my stomach nearly as much as cubensis do. So a whole lot less nausea and a much better, cleaner experience. And you have to eat a whole lot less mushroom for a similar effect.
Did I hear you say zapaticorum grows to 3 feet?
I said 1 foot.
1 foot. All right. Even 1 foot. That's a big ass mushroom.
yeah. And they're not always that tall, but in cultivation, it's not uncommon to see people with like a whole bunch that are all like 9 inches long. And in the wild, I've seen them definitely a foot, maybe 14 inches tall.
Wow. All right. So, still kind of hitting on this idea that we've been doing cubensis so far, because I want to encourage folks to break away from cubensis, because so many people that I talk to, regular enthusiasts, kind of think that all of these mushrooms that are in the hobby are all different, and it's surprising to find out that they're so similar. I've heard a mentor say to me that most of the modern strains that we're still using can really be traced back to a handful of original spore prints that were circulating during the 70s and 80s. Is that just lore, or does that sound pretty accurate to you?
That's probably true, but it's also really easy just to go out into nature and collect a fresh wild one. But I think it's true that 99% of what's cultivated comes from collections from a long time ago, because these kind of have stabilized genetics, and they've already been bred for more potency or a certain look. And so, yeah, they probably don't really have very much genetic diversity, but you could certainly go to Florida or eastern Texas and grab a wild cubensis and spore print that and cross it with one of these modern strains to introduce a bunch more genetic diversity. And that sounds really awesome, although people don't usually do it because it doesn't really seem to benefit the cultivator in any way, other than just kind of like throwing a wild card, giving yourself the chance of getting a strain that's significantly different than its parents.
I see. So there really isn't any advantage to freshening up the genetics if they're just the same. If you happen to hit the lottery and get a variety that's atypical, well, that's certainly going to be a win. But if you're just looking to refresh the old print, there's not really much advantage to it.
Not with Psilocybe cubensis. And some species go senescent, and senescence is when the DNA keeps growing and growing and basically gets tired and old. And that happens with some species a whole lot more than others. Like with Cordyceps militaris, it happens very quickly. And with oyster mushrooms, it basically never happens. And cubensis is a little bit more like oysters. It's kind of hard to know. It's kind of hard to measure senescence because there's so many other variables. But in general, there doesn't seem to be a whole lot of benefit from going to a wild print and getting all of these different genotypes in the DNA.
This is kind of a stretch of a question, but I'm going to go for it anyway. You're talking about this the genetics kind of losing its moxie after a while, and we certainly run into this a lot also in cannabis, right? When we're making clones of clones of clones, the plant that we might be growing now while it's acting fresh and young for us, it might actually be 20 years old itself. Is that kind of what happens in mushrooms too? I've never really thought of how like age and maturity works in mushrooms, but do they eventually kind of like lose their inertia as well?
Some species much more than others, and cubensis doesn't seem to do it much. Another
probably good reason why it was leaned on for universal use and acceptance. Yeah.
Yeah, it doesn't really peter out like some other ones might.
So as I was getting prepared to talk to you, I was learning about cubensis, and I came across the debate about whether or not cubensis is really going to stay one species or not, or rather if it's a species complex. And as I understand that is a group of species that are presently together, but when they're looked at in more detail, they'll find important ways that they tease apart. Do you think that cubensis is going to end up being a species complex and broken apart eventually?
Yeah, and it already is a species complex. And so cubensis is named after Cuba, where it was discovered, but it's actually an Asian species. And so it comes from Asia or Africa. And if you go over to Asia and Africa, there's a bunch of species that look very similar and are closely related. So it's already a species complex over there. And every species on Earth is moving apart, just like all the galaxies in the universe are moving apart. So everything is speciating, and in fungi, no one really knows what a species is. It's pretty amorphous, and so that's kind of an interesting thing. But biological organisms just do that over time. They never stay the same. They're always changing and adapting and mutating, and so the cubensis that we have is mostly one species, but there's a couple of them, like natal superstrain and Transky, are actually Psilocybe ocratiocentrata, which is pretty common in South Africa and Kenya, places like that. And that one has only recently been recognized as its own species. But if you sequence the DNA, you can tell it apart pretty easily. And it has pretty different effects than a regular cubensis, and almost everybody likes it better. So if you want to grow something a little bit different, but don't want to go like to a completely different thing, that's a pretty good option. Is just get a natal super strength or Transky print, and everybody that tries them says that they really like the experience of those, and they're pretty strong compared to your standard cube.
I heard a little bit of hope in there, and I want to pull that out, because I think we can all use as much of that as we can today. You made this great simile between the speciation of mushrooms to be akin to the expansion of the universe, and most of the time we are focusing on how humans on Earth are destroying the environment and limiting species and moving species more towards extinction, and the hope that I think I hear in here is a suggestion from you that mushrooms might actually be getting more numerous and more varied as time goes by. Did I interpret that correctly? Is that what you're saying? Because that's a cool thing if true.
Yeah, well, that's definitely true. Every organism on Earth is always splitting in different species, and it's really hard to say what a species is, because it takes thousands of years to make a different species. And the mushrooms don't really care what we call them; they just are kind of doing their thing. But their thing definitely involves getting more diverse. And it's not just mushrooms; all the plants and bacteria, everything on Earth is getting more and more diverse as time goes on.
So I used to be really focused on freshwater animals and plant species, and I had 35 aquariums. I was really into it, and it blew my mind when suddenly the taxonomy for the shrimp that I had been breeding for sale was changed from Neocaridina heteropoda to Neocaridina davidi, and I was all like, "What the hell? Like, can they do that?" And then I realized that this actually happened with Psilocybe too, where it got broken up, and half of the mushrooms got renamed. For those of us who are not on the inside of how that played out, would you just kind of tell us the story of how that happened? Because I found it pretty interesting.
Yeah, so every genus has a type species, and that type species is the official representative of that genus. And so a long time ago, we used to figure out what was related to each other just based on what things looked like and the microscopic features. And within the past 25 years, all of a sudden, we've been able to DNA barcode things. And with this DNA becoming cheap and accessible, all of a sudden, we get some real insight into what's related to what. And it turns out that we were only right about half of the time. And the other half of the time we were wrong, and that's due to convergent evolution, where two things that are not closely related have similar ecological pressures and end up looking real similar. But if you check the DNA, then you realize, oh, they actually arose from completely different places evolutionarily. And so that happens all the time, not just with mushrooms, but with shrimp and plants and birds and, just about everything else. And so the type species of Psilocybe was Psilocybe montana, and has been that way for a very long time. over 100 years, and people were happy to call them that. And it turns out that half of the psilocybes didn't stain blue and didn't have any effect if you ate them, and the other half did stain blue and had effects when you eat them. And psilocybe montana was in that group that did not stain blue and didn't have any effect. So with the DNA barcoding, people realized actually these ones that don't stain blue are quite a bit different genetically from the ones that do. And if you make a phylogenetic tree, so that's a tree of life, you can see that there's a whole bunch of other mushrooms that are in between them. And so genera need to be monophyletic, which means that they all arise from a common ancestor. So you can't have a genus that has a whole bunch of things from different branches of the tree of life all crammed together into one genus. So that presented a problem in psilocybe because the type species of psilocybe was Psilocybe montana, and that meant that all of the bluing ones needed to have a different name than all of the non-bluing ones. And so the type species of psilocybe was Psilocybe warora, which is from New Zealand, and that's that bluing puffball that you find on tree ferns there. And then the type species of type species was Montana, but basically it looked like what had to have happened was that all of the psilocybe that were bluing needed a new name. And so what they did, and by they, it's Gaston Guzman and a few of his collaborators, is they submitted a proposal to the International Nomenclature Committee, and they proposed that they change the type species of psilocybe to Psilocybe semilanceata, the Liberty Cap. And then they used the name Deconica for all the other ones. And so that was a good idea, so we didn't have to rename all of our psilocybes to a different genus. That would have been unfortunate since they have psilocybin and everything. So now the Deconicas are their own group, kind of closer to Galerina, and the psilocybes are retain the psilocybe name, and that kind of added some nomenclatural stability to all of this. But yeah, that was done about 2014, and I think that was a good idea.
It's interesting too that I was understanding that the way taxonomy works, the name actually, some were arguing, should stay with the non-psychoactive mushrooms. But of course, it'd be bizarre to have the psilocybe name with the letters for psilocybin in it, describing the non-hallucinogenic mushrooms. It seemed the way that I read it sounded like they decided, okay, we'll let the psilocybe stay with the hallucinogenic mushrooms, just kind of like as an administrative decision, because changing all of the science and all the people who are into it, it just seemed like a monumental move. Do I have a good read on that they just like it was a non-science, more administrative decision because it just made more sense as far as like study usability?
Yeah, that's exactly right. And so this is done through a proposal to conserve the name. And I just pulled it up here. It was published in 2007. So in 2007, it was Scott Redhead and a few other mycologists, the Mexican mycologists, Laura Guzman and Gaston Guzman. And they all wrote this little proposal. And so they're basically asking for an exception to the nomenclature rules, and that was accepted. And so kind of dodged that bullet back in 2007.
Does the mycology community at that level, I mean, from being involved with aquariums and cannabis as much as I have been, I understand how there can be full contact drama at these high levels of science. But did a move like this happen relatively undramatically, or was it messy at the time and it's easier to look back at it than to have lived through it?
I think everyone was just relieved that it was accepted and that we didn't have to make all those name changes. But, the mycologists generally get along pretty well. You don't have a whole lot of drama. And one reason for that is that there's really there's no money in mushrooms. And so with something like the cannabis world where there's a whole lot of money involved, it causes all sorts of drama. But with mushrooms, it's kind of the opposite, where there's almost no money in it. Kind of a mixed blessing, because it keeps all the people that just care about money away. So the only people that study mushrooms are people that actually care about them. And you don't have disputes over money. But it also means there's really no money for research. So there's tons of new species of mushrooms that get discovered every day. And very few people are actually publishing them and writing up the new species.
It's amazing how in just common conversation with people about mushrooms, how many people think that, the mushrooms that are to be discovered have been discovered. And if there is any kind of discovery, you're going to need to go to some super exotic place, at the equator or something. But the more I have listened to you, there are new mushrooms everywhere. And I think it'd be really helpful to people to understand that more.
Yeah, there really are. In fact, if you go out just hiking in the woods anywhere in the United States, and if it's been raining really well and the season's good, you'll discover 50 to 100 new species per day. And they're new in that they're unknown to science, so there's no scientific name for them. They haven't been formally described, but we see them all the time, and some of them are extremely common. Some of the most common edibles in grocery stores, some of the most common mushrooms that you might see 1,000 times a day. Even those don't have names yet.
So that's a really incredible, claim that there's so many, even where I live in the Pacific Northwest, we do have a lot of mushrooms here, but still, that I could go out and find so many just walking. Do you think that they are undescribed and unknown to science simply because of what we were saying earlier, that there's not really money in it for people to go out and spend their time that way?
Yeah, that's exactly it. And it's not like they're unknown. I mean, if you get a field guide to the Pacific Northwest, every mushroom will be in there and they'll all have names attached to them. They just won't be the right names. Oh. So there's two ways we deal with that. One way is that we misapply names. And so we will borrow a name from Europe and apply it to our mushroom, even though we know it's different because we don't have a name. And that's extremely common. If you just flip through a field guide, probably half of the names in there are misapplied European names. And the other way we deal with that is adding temporary names. So whenever I sequence the DNA of a mushroom and it doesn't come back as something that is described that has a name, I will assign it a temporary name. And that name gets used on citizen science websites like iNaturalist and Mushroom Observer, and even written about in scientific papers. That way, at least we have something to call all of the names. And these names might be something like Entoloma species California 82. And that's because we have 82 species of Entoloma that we have sequenced in California that we don't have names for. And most of those are probably unknown to science. So that is a whole lifetime's worth of work right there for somebody. But you can also apply a provisional name. For example, you have Psilocybe gandalfiana, and that is an unpublished name, and it's going to be published in the future. But for now, it's just a temporary name. But at least when we see it, we can communicate to other people about what it is that we found. And it's really important to have names for mushrooms for two reasons. One of them is so we can talk about what we found. And when you're doing research on a mushroom, so you're going to do a chemical analysis, you have to be able to communicate exactly which mushroom you analyzed. And so if you read any of the older papers on the chemistry of fungi, they'll be using the wrong name. And so they'll say that they tested one mushroom, but they actually tested a different mushroom, and these will be different chemically. The other reason it's important to name fungi is for conservation, because if something is very rare, you have to have a name for it. So you can say, oh, we have to save this really rare thing. It doesn't really work to be like, oh, we got to save all of these unnamed things. But if you find a mushroom, you sequence the DNA, and it does not match anything in GenBank or any of the other DNA databases, then you can say, oh, this mushroom only occurs in this one little forest. And, maybe it actually occurs other places, but if it's only known from one place, then you can say, well, you probably shouldn't cut down this forest because maybe it goes extinct if you cut it down. Maybe there's something special about this tract of land here. But there are so many unnamed mushrooms that you could literally go into any forest anywhere in the world, collect a whole bunch of fungi, and find novel DNA barcodes there. And that's kind of a cool thing, about fungi, and just we're just starting to learn about all of them. But really, just a very small percentage of the fungi are named. Probably between 1 and 10% of the fungi out there are named, and the rest of them do not have names. And so it's more likely to have a name if it's big, or if it's psychoactive, or there's some reason for people to pay attention to it. Most of the fungi in the world don't make mushrooms, and but even most of the mushrooms still remain unnamed.
That's startling. Everything you just said there is very startling. The fact that so few of them have been described properly, and that it's inspiring too that there's so much exploring left to do. I've got to hit on that part though, that so many of them are misnamed, and that European names have been brought over and misapplied, and so many of the guidebooks are somewhere between vague and wrong. When they're in the book and it's all made to look so orderly, it feels like science, but to find out that many of them are misnamed is surprising. What do you recommend for people who want to make sure that they are learning accurate taxonomy and I guess to some degree nomenclature for these mushrooms? Is there does everybody kind of have to start with these early books that have got so many inaccuracies, and then over time they weed them out, or do you recommend for people getting into mushrooms to like skip that whole consumer layer of books because of the inaccuracies and start somewhere else with a more scientific based book?
Well, there really are no books that have all of the updated names, because as soon as you publish a book, it's a snapshot in time, and the names keep changing, and as our understanding keeps evolving. But there's certainly a newer book is going to have less of a learning curve, as when you try to use the newest, best name as opposed to an older name. But older books still work fine. Also, old names or misapplied names, the wrong name is still way better than no name. If somebody uses a name from 100 years ago, I'll still know what they're talking about. It's just that maybe we have a better name now that more accurately reflects its true genetic relationship.
And I guess you might have a name from 100 years ago that might not be the right name anymore, but at least what that name, the four of them that name was broken into. And so you go, all right, I'm going to go look at these four.
Yeah, exactly. Mm-hmm. And a long time ago, almost every guild mushroom was an agaricus. So Amanita muscaria was named Agaricus muscarius, and old scientific names are still valid. So you can still call it, Agaricus muscaricus, and that's not wrong. It's just that we have a more updated name now that more people know.
Right on. All right. So we're going to take a short break and be right back. And when we come back, we're going to start with a conversation about the kinds of pressure that you need for mushrooms to evolve. You are listening to Shaping Fire, and my guest today is mycologist Alan Rockefeller. And, you know, without these advertisers, Shaping Fire would not happen. So please support them and let you know that you heard them on Shaping Fire. The new finely screened living compost from Leighton Morrison at Kingdom Aquaponics is in a product category all by itself. Nearly all compost you can buy is thermophilic, meaning that the compost pile has been heated in order to destroy all the pathogens. This composting process leaves you with only thermophilic organisms while killing everything else. Living compost from Kingdom Aquaponics is never heated above 115 degrees, so it still thrives with the phosphate and potassium solubilizers, degraders, and decomposers, along with the more advanced nutrient cycling organisms. But you might be thinking, Shango, if the compost is never heated, isn't it filled with pathogens that might hurt my garden? All of Leighton's compost components are specifically chosen and pre-processed to remove pathogens before the cool mesophilic composting process. The mushroom substrate he uses is sterilized in advance for growing mushrooms. The wood fines are from shredded kiln-dried lumber, and that kiln heat removes any pathogens. And the repurposed coco coir is sterilized with heat in advance too. These materials are then combined with a mix of worm castings, fish poop, and special biological inoculants that Leighton incubates and blends himself. The resulting compost has biological stability, structure and water storage capacity, is packed with nutrient cycling protozoa and nematodes, and is fungal and bacteria rich, including purple sulfur bacteria. He even provides the biological reports. This ultra-premium living compost is for anyone who wants to make compost teas, extracts, or top dress. You're getting 3 ingredients in one: worm castings, fish mulm, and biologically active compost all in one product. Get it right the first time. Buy living compost now at kingdomaquaponicsllc.com. Available in 3 convenient sizes. That's kingdomaquaponicsllc.com. This message is for folks who grow cannabis. I'm talking to home growers, patients, and commercial growers too. I'm probably talking to you. When you plan out your next growing cycle, be sure to check out Humboldt CSI seeds at humboldtcsi.com. Caleb Inspecta and his family have lived in Humboldt County for over 100 years. For the last 40 years, 3 generations of his family have cultivated extraordinary Sinsemilla cannabis in Humboldt, Mendocino, and Trinity counties. Because of his lineage and the hard-earned experience that comes from growing up, smoking, and sifting large populations of cannabis plants in Northern California, the seeds you'll cop from CSI will be winning genetics based on longtime heavy hitters and updated and resifted to bring out new and exotic traits and better yields. Go ahead and ask around. Caleb, also known as Inspecta and Pirates of the Emerald Triangle, is a breeder's breeder. He reaches way back and works with significant strains, recreating them in new and interesting ways that you'll love as a toker and a grower, as well as offering you some surprises that will delight serious seed traders and cultivators. Humboldt CSI goes a further step and selfs all these chemovars so you know all the seeds will be female. These are not experimental feminized seeds. Humboldt CSI releases some of the best female seeds available anywhere, and it will show in your garden. Folks grew quite a bit of CSI Humboldt genetics last year here on Vashon Island, and everyone was pleased. The patients had beautiful female plants and didn't have to cull half of their garden as males. The folks growing for the fun of getting high grew colorful flowers with exceptional bag appeal and great highs. And breeders had 7 out of 7 females in a pack, which gave them a lot of phenotypic choices. Take a moment right now and visit humboldtcsi.com. You'll find an up-to-date menu of both feminized and regular lines, along with photos and descriptions. That's humboldtcsi.com. Businesses everywhere are striving to reach people through advertising. We all know, though, that trying to reach a cannabis audience with a quality message is pretty difficult. That's why many people choose to advertise on the Shaping Fire podcast. Advertising on this show allows us time to talk about your product, service, or brand in a way that really lets people know what sets your company apart from the others. Bold people who own companies know that getting into relationship with their customers is essential, and that's what we offer. We will explain your service or product and what sets it apart as desirable and help our audience get in contact with you. It's pretty simple, really. Advertising does not have to be all whiz-bang smoke and mirrors. Nowadays, I find that people prefer to just be spoken to calmly, accurately, and with good intentions. If you want to make your own commercial spot, you can do that too. Most of the options for your advertising are dying in the modern media sphere, but podcast listening is skyrocketing. With a commercial on Shaping Fire, you'll reach your customers in the privacy of their headphones right now and will continue to reach new listeners as they explore the Shaping Fire back catalog of episodes again and again for years. And we won't work against you while taking your money like Instagram. A spot on Shaping Fire costs less than a printed postcard per person, and the Shaping Fire audience is full of smart cannabis enthusiasts, cultivators, and entrepreneurs who are always curious to learn. Do yourself a solid and contact us today for rates on podcast advertising. Email hotspot@shapingfire.com to find out more. Welcome back. You are listening to Shaping Fire. I am your host, Shango Los, and my guest today is mycologist Alan Rockefeller. So before the break, we were talking about how many different varieties there are still for us to describe, and all the room there is for folks to explore in the mushroom world. And Alan, these mushrooms, they continually change because of pressure from the environment, whether it be, weather or nutrition or even humans. Will you tell us a little bit about the arc of change? Like you mentioned earlier that this happens for over a long period of time because they don't work on the same clock as humans, but also over that kind of time, the pressures are different than the ones that we as humans experience. So will you illustrate that with us for a story or two about what you've seen influence mushroom evolution?
Yeah, so that's one of the cool things about mushrooms is that the evolutionary pressures are so different than they are with plants or animals. And so that makes plants and animals very easy to understand because they all, the plants have their pollinators, the animals have their predators. But with mushrooms, the evolutionary pressures are mostly things that we don't know anything about and that we can really just not even comprehend. And so that makes mushrooms very mysterious. And that's probably the reason I'm so drawn to them, is that they are just really mysterious. And, I always tell people anytime you have a question about mushrooms that starts with the word why, the answer is always the same. I don't know. Nobody knows. And so we have so much to learn about mushrooms. And yes, those evolutionary pressures that are shaping them, but most of those evolutionary pressures are things that happen deep underground or inside of a log. So we just don't have any idea what's going on, but it's definitely chemical warfare with other species of fungi and bacteria, viruses. And, we know that they like rain, so some mushrooms will adapt to drier areas by maybe the cap not opening up so much. So it's more like a puffball on a stick. We see that a lot in more arid areas. But for the most part, we just have no concept of what these pressures are. And the evolution has been really interesting. You have all of these crazy chemicals, all of these pigments. And you have mushrooms that are almost any color of the rainbow, but nobody really has any idea why any mushroom is any color, whether it has a benefit to the mushroom or not. If it does have a benefit, how does that work? It's just completely mysterious.
Well, while we're talking about color, I was going to ask you this during the third set, but let's put it in here. The mushrooms that are commonly called albinos, but are usually leucistic in the modern cultivation world, they're very wanted. Like the albino penis envy and others, albino Louisiana. And is any of is there an attractive aspect to the leucistic part about its functioning, or is it just because a leucistic mushroom just looks cool because it's so different? Is there anything biologically about it that makes it more preferable than the non-leucistic variety?
Well, a lot of those albino strains do tend to be pretty strong, so maybe they're putting more of their energy into producing alkaloids since they don't have to produce all of the color pigments. But that's just a guess. It seems like these pigments are missing occasionally just by chance, and that can happen in any mushroom. Just sometimes, like 1 in 1,000, you'll come across a mushroom that is just missing its color, and typically there's several different molecules that all mix together to make the color of a mushroom, so usually it's just missing one of them. And so for example, the spores often have a dark color, and often the darkest color is missing, giving spores kind of a reddish look, so that'll be the red spore varieties. But sometimes other pigments are missing and you get other colors, and then sometimes all of them are missing and the spores just end up being white or clear in a normally dark spored mushroom. I think with the spore color, that's one pressure that we kind of figured out what that does do, because the ultraviolet light, the UVA and UVB from the sun sterilizes, breaks the DNA chain, sterilizes the spores. So it's more likely that dark spored mushrooms are found with a wider distribution. Something like Coprinellus micaceus or some of these Sapphirellas, you'll find the same mushroom all over the world, whereas with a white spored mushroom, it's much less common that you find the same exact species on different continents. And that's probably because when you have a white spored mushroom, any ultraviolet that hits it will just sterilize the DNA in there and kill the spore.
Wow. Developing their own suntan lotion. One of the wildest discoveries that I've come across in mushroom science was that the genes responsible for psilocybin seem to have jumped between species. And I learned this term for horizontal gene transfer, which again puts us kind of in the realm of things we don't know and magic, I guess. But could you explain what we know about horizontal gene transfer in fungi and what that tells us about the evolutionary role of psilocybin in these mushrooms?
Yeah, so normally in evolution, you get your genes from your parents and, things get passed down that way. But occasionally in nature, you find these genes that are not from the parents and they're just completely different. And so you'll find genes from a completely different organism inside of another organism, and that's called a horizontal gene transfer. And there's a lot of ways that can happen. There's bacteria and there are viruses that will insert foreign DNA from one organism into another. That's how Monsanto makes all of their transgenic crops, as a bacteria called Agrobacterium. But it also can happen just due to physical damage. Like if you take two completely different types of mushrooms, put them in a blender and blend them up, the DNA will break, and then it'll try to repair itself. And when it does that, it'll just try to find the broken ends and stick back together. And sometimes DNA from completely unrelated organisms will stick together, and occasionally it continues to function. And so this psilocybin gene cluster got inherited in psilocybe about 30 million years ago, or got inserted with horizontal gene transfer, and that's quite a while ago. Humans have only been on the earth about 300,000 years, so these psilocybin mushrooms are much older than humans. And so we can only guess as to how and why that happened. But just looking at all of the DNA sequences, we can kind of figure out when these organisms acquired these genes. And I think it's actually pretty common common in nature that horizontal gene transfer happens. It's just that usually it's not in such an iconic thing as the psilocybin gene cluster, so we usually don't notice it. But there's a lot of different mushrooms that have psilocybin, and they are not all closely related. You have 6 or 7 genera, and the psilocybin gene cluster for most of them is the same gene. And so they definitely did not evolve independently. Psilocybin evolved in something and then got transferred horizontally to all of these different mushrooms.
But we don't know what that was.
No, we don't know what it is. There's a paper by Jason Slott where he found some similar genes in the Atheliales, and that is a group of crust fungi. So it's possible that they got the gene from some crust fungi, but it's also possible it came from somewhere else. Maybe it came from something that went extinct, and now these genes live on in the mushrooms, but the original organism that evolved them is no longer here.
So when I was reading about horizontal gene transfer, I was thinking it was more about mushroom to mushroom, different varieties reaching each other with their spores. But now I realize it's much bigger than that. Do you think that we could be still experiencing horizontal gene transfer regularly now, but we don't know it because it's going to take tens of thousands or hundreds of thousands of years for it to evolve? Like somehow it always seems like all of these crosses happened, all these millions of years ago. But based on what you're saying, they could also be happening right now, but we're never going to be around to see them.
Yeah, well, the part that's slow is the evolution, the mutations over time, and a beneficial mutation doing better than another one. But horizontal gene transfer is a very rapid thing. It happens just, basically instantly. And so you just all of a sudden have these genes transferred from one organism to another. And I think it probably happens all the time in nature. It probably happens inside your body every day, and you just don't even notice it. But it's just, I think it's rare that we do notice it.
So when we were talking in the first set about how many of the mushroom species are yet to be discovered, and that you are out in the forest and elsewhere finding new psilocybin species yourself along with others. I know this is a very speculative question, and luckily it doesn't start with the word why, but do you think that we've even found half of the psilocybin expressing mushrooms out there? And of the ones that are still to be found, do you think that they are, clustered in some part of the world because of the environment that seems to be more friendly to psilocybe? Or do you think that they're just as likely to be anywhere at this point, just because they've had so much time to spread around?
Well, I think we've probably discovered about half of them. And certainly a lot of the bigger, more common ones that are found in places where a lot of people live are more likely to be named. But even in a state like California that has people all over it, there's new ones being discovered here. There's probably new species of psilocybin mushrooms to be discovered in every state. And then some groups are just studied a whole lot less than others. For example, psilocybe is generally pretty well studied because they're so iconic, but Gymnopilus and Paniolus are 2 other genera that are extremely common, much more common than psilocybe, and they are kind of a taxonomic mess in that some species were described, but we don't really know which names go with which species, and there's a whole bunch of them that just don't seem to have names. And so, a lot of in North America where there's tons of people around, a lot of the new psilocybin discoveries are going to come from Paniolus, Gymnopilus, Conoscibula, Inocybe, some of these other genera that people don't pay quite as much attention to.
We've been talking. Go ahead.
The second part of your question, are there some parts of the world that are more conducive to psilocybin mushrooms? And, that's really the cloud forest habitat. And so cloud forest is something that we don't get in the United States, but you don't have to go very far south of the border before you start getting cloud forests in Mexico. The furthest north is a place called El Cielo in Tamaulipas, just a little bit south of Texas. And then as you go further south in Mexico, you get more and more cloud forests. And the cloud forest is one of the most diverse ecosystems in the world. And it's something that you see between 1,000 meters and 2,500 meters elevation. And these forests get a whole lot of rain. And then most afternoons, the clouds come in. Very often it rains kind of every afternoon. And so you get a lot of precipitation there, a lot of humidity. And if you go down in elevation, it starts to get real hot and things dry out quicker. So then you get into the tropical ecosystems. And then if you go up in elevation, it starts to get very cold. And so in these tropical ecosystems, the mushrooms that tend to do well are these smaller white spored mushrooms. And as you go up and it gets colder, the mushrooms that tend to do well are larger. So that's where you find a lot of your really big porcini and chanterelles up at the high elevations. But in the middle, you have the highest biodiversity, and you go into these cloud forests, and the plant diversity is just insane. You have hundreds of species of trees per acre, whereas here on the west coast of the United States, we might have 5 or 10 species of trees per acre. And the fungal diversity follows the plant diversity. And then if you start looking at the mushrooms, you'll see cordyceps everywhere, just thousands of different kinds. And those are the habitats that you find the most diversity of psilocybin mushrooms. Just sometimes we'll find 10 different species of psilocybe in one day in some of these places, just because the habitats are so diverse.
Just hearing you describe that space is so inspiring. I mean, it sounds like a science and photography wonderland. It sounds really wonderful. I'm curious whether or not these cloud forests that have got all of this very delicate biology, are they starting to get trampled by folks around the world who are interested in these and other mushrooms, who are, looking to explore it? It sounds like a delicate environment. And if it's at the top of the list, I can imagine, citizen scientists and corporate scientists and university scientists and, backpackers and, all these people going to this area to see the beauty and to maybe become famous by, finding their specialty mushroom or something like that. And it makes me have concerns for the environment itself. Is it so early that we're not seeing that yet, or is the destruction of these mushroom habitats by curiosity seekers already having an impact?
So these cloud forest habitats are being destroyed at an increasing rate, but it's not the curiosity seekers that are doing it, it's the loggers. And so in Mexico, they've already logged 99% of the cloud forests. And so the logging continues at an increasing rate every year. In Central and South America, they get rid of a lot of them to have cattle and farm fields, that sort of thing. And so the impact of people that are curious and want to go collect mushrooms or collect plants is just about nothing compared to the impact of the loggers. And these mushrooms are a very sustainable resource. They've done studies where they pick every single mushroom in a study plot over 30 years. And what they find is that if you just leave all of the mushrooms there, or you rip all of the mushrooms out of the ground, or you carefully cut them off with a knife, there's really no impact on the number of mushrooms that come back the next year. But what does really impact them is cutting down the trees. So if you cut down the trees, then the mushrooms will go away, and you'll find just the same 40 boring mushrooms that you find in every suburban backyard in these clear cuts. And after about 100 years, then they start to come back. But if you put a parking lot instead of the forest, then they don't really ever come back. So it's really humans that are, destroying these ecosystems, and they just don't see the value in these ecosystems. And that is one of the cool things about mushrooms, because if you can teach people to forage mushrooms for food or for medicine, then they have a reason to go to the forest, and they have a reason to preserve these forests. People that live in the cities and just don't have any reason to go to the forest, they're not going to say anything if they propose to cut down one of the forests. But if you got a bunch of foragers in there that are looking for food and psilocybe and everything else, then they're going to complain if somebody proposes to cut down their local forest. And that's one of the reasons that it's important to get more people into nature, get more people into mushroom foraging. And because when people start picking mushrooms, they spend a lot more time in the woods, and they realize how cool and how interesting nature is, and how valuable these places are.
Yeah, support your non-local cloud forest t-shirts. We want to make them more popular so they're easier to protect.
Yeah, and the cloud forest habitat is the most biodiverse, but there's cool mushrooms in any habitat. Even the deserts outside of Los Angeles, California, have really cool mushrooms in them when it does rain. So these fungi are just waiting for the rains, and after the rains they come up, you get all these crazy puffballs on sticks and all sorts of things. So there are cool mushrooms in every habitat, and even ones that seem very desolate, like a dry grassland. When the rains come, you'll find really cool, unique mushrooms in the grasslands and in the chaparral that you won't find anywhere else, and that you wouldn't see in a really nice forest.
We've been talking about arrays of mushrooms and how many more there are to be described, and how many of the old descriptions are proving inaccurate. And, I think that most of this and most of our realization of this is due to DNA sequencing technology and how it's rewriting mushroom history. Would you think that that is right, that that's the key technology at the core of it? And also for folks who are not deep into this aspect, would you explain what DNA sequencing is doing for mycology as a whole?
Yeah, that's accurate. And, it's not just mycology, it's every branch of life has been revolutionized by DNA sequencing. And, DNA sequencing is really just reading the DNA. And so these molecules encode a huge amount of data, and they're so tiny that until recently, we just didn't have any way to read them. But starting in the 1950s, people started to figure out that this thing called DNA exists, and they started to have different ways of getting insight into what it's saying. And it wasn't until the 1970s that people were actually able to read the individual bases just like you'd read a book. And it started out being extremely slow and extremely expensive. And then in the late 90s, it started to all of a sudden get cheap and practical. And all of a sudden, even people in just like kind of like a medium level college laboratory could start reading the DNA of things. And since then, every year it gets cheaper and easier. And so now you can set up a DNA sequencing laboratory in your home for under $1,000. Oh. And start sequencing things. I'm sitting in my laboratory right now in my house, and I have a little DNA sequencing table set up over here. And so I got pipettes and a PCR machine and a whole bunch of chemicals, and I can just take any living thing. It doesn't even have to be a mushroom, and put it into a tube, grind it up, put it into a machine. And if I start the process about 9 in the morning, I can have my results by midnight. And so it's really incredible. And it's also enabled people to make really cool discoveries on their own. So there are not very many branches of science where just one person who is a beginner can make a really valuable discovery. But mycology is probably the science where it is easiest for beginners to make a huge impact. And so you can, you can sequence these things really easily, and it's actually cheaper and easier to do it at scale. So there's a laboratory called Mycology Lab run by Stephen Russell, and he sequences probably about 20,000 mushrooms per year. And he has events called mycoblitz where anybody in the world can just send in unlimited quantities of mushrooms, and he will sequence them for free. And so he gets grants and donations to fund it. But if you do it at scale, the price goes down to about 20 cents per DNA sequence.
Whoa.
So, yeah, you can discover new things very inexpensively. And a lot of places in North America have free DNA sequencing. I think you're up in or you're up near British Columbia.
Sure, 4 hours.
British Columbia is one of the places that has free DNA sequencing. And so you can, anybody that lives in British Columbia or Alberta or California, there's a whole map of places where you can get free DNA sequencing, or anybody can do it for free during the mycoblitz. But, people just doesn't really take any specialized knowledge at all, just to go out into the woods, take pictures of mushrooms, dry them out, and drop them in the mail. And then the DNA sequence pops up on your iNaturalist observation, and people will come along and identify it based on the DNA sequence. And so this is something that we really need people to do, because a DNA sequence by itself is meaningless. It doesn't have meaning until you compare it with other DNA sequences. And so the more that there are, the more meaning each one has, and the more we understand about the fungi in any given part of the world. And so, it's really important to have people just collecting and sending stuff in. And even here in California, we discover new novel sequences that no one has ever found before all the time. But in places that people do not collect mushrooms very often in, like northern Canada, North Dakota, South Dakota, Montana, Louisiana, places that don't have a very established mycology culture, there's not very many DNA sequences. I think we have maybe 1 or 2 from South Dakota and 0 from North Dakota. So we just have no idea what fungi are even there. And so, anybody can send these in, and also anybody can start naming these species. It doesn't take someone with a PhD or any college degree at all to name new species of mushrooms. All you have to do is find the thing, DNA sequence it, and then do the microscope work and write up a paper and publish it. So it's the sort of thing that anybody can do with really no training at all. And we need a lot more people doing this, because without it, it's going to take us many, many thousands of years just to sequence, or just to name even the common mushrooms that we see every day.
Wow, I cannot be the only person who is super inspired by that, Alan. Understanding, hearing it laid out like that, you make it sound so accessible. And my next question was actually going to be about, the bottleneck that I thought there was with the technology. I was going to say, all this interest, it's too bad it's bottleneck because the technology is so expensive. But, the fact that somebody can set up a basic lab to do this at home for under $1,000, it's just the opposite. This is empowering like citizen scientists to get involved, and there sounds like there's so much opportunity. Like I'm on fire. I want to go start running DNA on mushrooms myself, just because it sounds like such a curious thing to do. Are you seeing this happen around you, like where mycology is getting more participants and it's becoming more, I don't know, like even it's more accessible by regular folks who are not heavy duty degreed people with access to university money?
Yeah, absolutely. And, one thing that's really helped is the internet and social media, because now people can take a picture of a mushroom, post it online in places like, for example, the Mushroom Identification Forum on Facebook, or, really anywhere that people hang out online. You can post photos of mushrooms, and people who know what they are will identify them for you. But yeah, it's a sort of thing where everybody really should be doing it, and they would be if they knew how cool it was. And it's the coolest thing in the world when you find something and you get your DNA sequence back and you finally figure out exactly what it is, and if it's rare or if it's new, that sort of thing. And a lot of times you get a DNA sequence back for something that no one's ever sequenced before. You're the first person ever to see where it actually fits in with the rest of life. And that's the sort of thing that's very easy to do. I mean, even someone that doesn't know much about computers could figure out how to do it in just a few minutes. And so, yeah, it is super exciting. And I used to go around teaching people how to set up their own DNA lab, because you can just take the DNA lab, throw it in the car, and then set it up on a card table, like at a fungus fair or a mushroom festival. And so about 10 years ago, I started doing that, and I was teaching DNA sequencing classes all over the world. But I haven't been doing that as much recently because it makes less sense for people to set up their own lab and more sense for people to collect mushrooms and send them into a centralized lab. And there's a couple reasons for that, but one of them is that this technology called nanopore came along. And with nanopore sequencing, it's a little DNA sequencer about the size of a USB thumb drive, and you can sequence thousands of mushrooms at once on this little tiny device. And since the sequencer is right there, you get your results. You watch them come across the screen. But the thing is that it's kind of expensive to run the device. It costs a few $100s to run it, but each run you can sequence thousands of things. So all of a sudden the bottleneck is not the sequencing, but the collections, because nobody has hundreds of thousands of different collections of mushrooms laying around that need to be sequenced. So typically what people will do now is collect mushrooms and document them in the field and then just mail them all to a centralized point, and then that person will run nanopore, and that makes it a whole lot more efficient. And that's how people are able to sequence many thousands of different collections per year.
Wow, that's fantastic. And I understand, you can do your basic stuff at home, but if everybody's really wanting the nanopore, well, then let's just put them all together. Also, everybody bringing their DNA to these shops that have got this technology, it helps keep them going too, because we don't want them to go away because people are doing a lesser variety test at home. We really do want to get as much as we can, all the data points we can.
Yeah, for sure. There's a couple reasons that I still sequence at home occasionally. One of them is that I get results pretty quick, because if you send them out to a lab, they put it together in a big batch and you might get your results in a couple months. Whereas if you do it at home, you can get your results in the same day. Also, if you send it into one of these big labs, they're going to be sequencing the same gene. And that's generally good, because it's generally just one gene called the ITS, the internal transcribe spacer, that you can use to identify about 98% of fungi. But occasionally you'll want to sequence any gene, and you can sequence any gene in the whole genome for about the same cost. And so for people that are doing this at home, they'll be using a sequencing technology called Sanger sequencing. And with Sanger, you just take the mushroom, grind it up, put it into a PCR machine for a couple hours with these two primers, and the primers that you select will choose which gene that ends up getting sequenced. So you can pick a primer for anything. You could take a human DNA and then put a primer in there that would sequence the gene for cancer or for eye color or anything else. Or with mushrooms, you can sequence the ITS gene if you want, but you can also sequence much more obscure genes, really anything. And so if I want to do a multi-gene analysis where I'm analyzing several genes instead of just one, I will run PCR at home and sequence 5 or 6 different genes of the same mushroom. And then I can get that done with the Sanger sequencing. And the way Sanger works is that the machine is pretty expensive, but there's a lot of labs that do it. So people will typically run PCR at home, and that heats up the DNA and cools it down a whole bunch of times until you have a very concentrated little droplet of DNA and then just drop it in the mail to a Sanger lab. And they typically charge about $5 per sample. And so when you're sequencing at home, you're paying about $5 per mushroom that you want to sequence. And that's fine for small runs. But if you have thousands of mushrooms, it ends up getting really expensive. And that's where nanopore ends up being the right way to do it.
Right on. All right, so let's go ahead and take our second short break, and we'll be right back. You are listening to Shaping Fire, and my guest today is mycologist Alan Rockefeller. After you've caught up on the latest Shaping Fire episodes, do you sometimes wish there was more cannabis education available to learn? Well, we got you. Shaping Fire has a fabulous YouTube channel with content not found on the podcast. When I attend conventions to speak or moderate panels, I always record them and bring the content home for you to watch. The Shango Los YouTube channel has world-class speakers including Zoe Sigmund's lecture, Understanding Your Endocannabinoid System, Kevin Jodrey of Wonderland Nursery talking about breeding cannabis for the best terpene profile, Frenchie Canoli's Lost Art of the Hashishin presentation, Nicholas Mahmood on regenerative and polyculture cannabis growing, Dr. Sunil Agrawal on the history of cannabis medicine around the world, Eric Vlosky and Josh Rutherford on solventless extraction, and Jeff Lowenfels on the soil food web. There are several presentations from Dr. Ethan Russo on terpenes and the endocannabinoid system too. While there, be sure to check out the 3 10-part Shaping Fire Sessions series, one with Kevin Jodrey, one with Dr. Ethan Russo, and one with Jeff Lowenfels. And even my own presentations on how to approach finding your dream job in cannabis and why we choose cannabis business even though the risks are so high. As of today, there's over 200 videos that you can check out for free. So go to youtube.com/Shango Los or click on the link in the newsletter. As consumers become more educated and regulations become more demanding, it is increasingly important to avoid the use of chemical pesticides when cultivating cannabis. Beneficial insects have been used for decades by the greenhouse, vegetable, and ornamental plant industry, and today many cannabis cultivators are moving from sprays and chemicals to beneficial insects. Natural Enemies has the beneficial insects, mites and nematodes, microbials, sticky cards, and air distribution units you need to partner with nature to defend your garden. Whether you manage acres of canopy or have a simple grow tent in your home, Natural Enemies is ready to help answer your questions and help you transition away from chemical sprays towards clean and natural solutions. Natural Enemies was founded in the cannabis industry and continues to provide cannabis-specific guidance. Natural Enemies beneficial insects are grown with care and precision by Copert Biological Pest Control. Since 1967, Copert has assisted growers in identifying pests and devising reliable solutions while providing healthy insects and mites that will protect your yield. Since the 1990s, Copert has been a leader in cannabis pest and disease control worldwide and have highly trained consultants to assist you in Canada and the United States from coast to coast. Natural Enemies and Copert can help. Visit naturalenemies.com and get detailed information. You know getting away from pesticides is good for health and good for business, and Natural Enemies is ready to help. Visit naturalenemies.com today. Sometimes the topics I want to share with you are far too brief for an entire Shaping Fire episode. In those instances, I post them to Instagram. I invite you to follow my 2 Instagram profiles and participate online. The Shaping Fire Instagram has follow-up posts to Shaping Fire episodes, growing and processing best practices, product trials, and, of course, gorgeous flower photos. The Shango Los Instagram follows my travels on cannabis garden tours, my successes and failures in my own garden, insights and best practices from personal grows everywhere, and always gorgeous flower photos. On both profiles, the emphasis is on sharing what I've learned in a way that you can replicate it in your own garden, your own hash lab, or for your own cannabinopathic health. So I encourage you to follow at Shaping Fire and at Shango Los and join our online community on Instagram. Welcome back. You are listening to Shaping Fire. I am your host, Shango Los, and my guest today is mycologist Alan Rockefeller. So, Alan, we've been talking about the value of not only DNA sequencing to understanding what's in the mushrooms, but also the variety of mushrooms and all of the important biochemicals and all the good healing that we can bring out of these mushrooms. But first, we have to find them. We know that these second metabolites in mushrooms provide important chemistry. And we know this at the very least because single molecule psilocybin studies really seem to be so much less effective for humans. So what do you think we're going to find the benefits are when we really start to understand the nature of these less prolific, but nonetheless really important biochemicals that are second to psilocybin, but still there and important?
Yeah, so psilocybin mushrooms have a lot of different chemicals in them that could be active. You got about a dozen different tryptamines, and those, psilocybin and psilocin are the most well known, but you have the baeocystin, aruginacin, and then a whole bunch of other ones that are there in smaller quantities. But some of these other ones also bind to the 5-HT2 receptors. And so they probably influence the effect of the mushrooms in their own way. And whenever you test different mushrooms, you'll find different ratios of all of these chemicals. So it's not like you find certain chemicals that are in one mushroom or another, but for example, psilocybe semilanceata tends to have really high levels of baeocystin, sometimes as much baeocystin as psilocybin, whereas baeocystin is usually just a trace chemical in psilocybin mushrooms. So every psilocybin mushroom has some baeocystin, but some much more than others. And all these other alkaloids are the same way. So with psilocybin, that one is not active on its own, but when you ingest it, then your liver knocks off the phosphorus atom and it becomes psilocin, and that does go through the blood-brain barrier. So that makes it become psychoactive. And all of these other tryptamines also get converted in your body into different molecules. And some of those are as or even more psychoactive than psilocin. A study came out just the other day that they found that aruginacin breaks down into 4-HO-TMT, and that one binds to the 5-HT receptors quite strongly. So that could definitely influence the effect that you get when you eat a mushroom. And then about 7 years ago, a lab in Germany, the Hofmünster lab, they found that mushrooms contain beta-carbolines. And so those are molecules like harmine and harmaline, and those are famous because they are the active ingredient in the ayahuasca vine that make DMT orally active. So it was kind of surprising to find these molecules in mushrooms. And it turns out that psilocybe cubensis has almost none of these molecules, just trace amounts in the fruiting body, but in the mycelium, you have quite a bit more. And then different species have completely different, very different levels of these beta-carbolines. But what the beta-carbolines do is they knock out the MAO enzyme in your body, which is an enzyme that gets rid of a lot of these molecules. So it makes the effects different and longer lasting of a lot of these tryptamines. And then even more recently, it's been discovered that psilocybin mushrooms have terpenes in them. And so there's a whole bunch of different terpenes. Paper came out also from the Hofmünster lab a couple of years ago, and they named some of the terpenes after the mushrooms. So you have like cubene is one that they first discovered in psilocybe cubensis. But they have about 20 different terpenes that they've detected. And those are kind of a mystery as to what they do. They probably do influence the quality of the trip in some way, but I think people know a bit more about what they do in cannabis than what they do in mushrooms. And then if you just take a mushroom and you run it through an LCMS, so LCMS is liquid chromatography mass spec, and a good LCMS could cost $500,000, so they're a bit expensive. But it's a really cool device because you can just put a mushroom in there and see all of the chemicals that are in there. And sometimes people are able to get them at a pretty good price from like a cannabis lab that shuts down or something like that. And that kind of equipment is what's used to figure out what all these molecules are. But you put any mushroom into an LCMS and you will see hundreds of different peaks on the graph. And some of them are known molecules and others are molecules that people have never really interacted with before. And you got to concentrate them and use NMR to figure out the exact structure of them. But there's just all sorts of different opportunities for these molecules to influence, different things in your body.
How sophisticated is our list of these secondary biochemicals? I can imagine that on one hand, they are not as researched as we want because there are so many mushrooms that we're not aware of. So this list would have to be, adolescent as well. But at the same time, it's also where I'm assuming that the pharmaceutical companies are looking. And so if there is money in research for this, it's probably at these. But then there's a third hand, which is these companies could very well be keeping all this information private. So what's the state of the science? Are we seeing a pretty comprehensive list of these chemicals that are in these mushrooms, or is it really early and maybe even politically motivated?
It's pretty early. There's all sorts of different molecules, and the tryptamines are pretty well characterized in that we know to look for them. So even somebody that just has an HPLC will have reference standards for the different tryptamines, and they will be able to figure out which tryptamines they have. But some of these other molecules, people don't really know to look for them. And we also don't understand what the effects are of these different tryptamines.
Right. And what their effects are is kind of at the corner of why everybody's looking in it for future medicine. One of the questions that always occurs to me is, if the mushroom makes psilocybin and then we convert it into psilocin, and then also psilocin occurs in the mushroom and in the wild, do we know why? We're back to why again. Do we know why or see any reason for the mushroom to also have psilocin? Because I understand that if we dephosphate it when we eat it, that's one thing. That's something that our body is doing. But to have both the phosphated and unphosphated version in the mushroom, that seems different to me. It's like it's partially digested for us.
It's a really good question. And, nobody knows why it's in there, but people have been trying to figure that out for a long time. And I think the person that's done more research than anybody else in that realm is Dr. Jason Slott. And he's at the University of Ohio. Really cool guy, great teacher. And he has written a bunch of papers that speculate why these mushrooms might have psilocybin. And it's really hard to come to an exact conclusion, but there's a lot of observations that we can make. So the psilocybin itself is a very stable molecule and doesn't tend to break down. But then these mushrooms have enzymes that dephosphorylate the psilocybin. So they just break the phosphorus off, and then it becomes psilocin, and psilocin is much less stable. And then there's more enzymes that take that psilocin and polymerize it into these long chains. And those long polymers have different colors. So that's where you get your blue staining, but you can also get pink and browns in those psilocin polymers. And so one theory is that the idea is that when you have another thing about these is that if you have psilocin in a mushroom, there's other enzymes that rephosphorylate it and turn it back into psilocybin. So that's why mushrooms are not blue when they're just sitting there. But then when you damage them, these enzymes kick in and they will dephosphorylate the psilocybin and then polymerize them. So that changes the color of the mushroom. So this whole pathway seems to be very intentional. There's a whole lot of enzymes that are working together to create this whole chemical pathway and to kind of change the molecules around. So it's definitely not random, not an accident, but why and what they are doing is more difficult. But, one reason that you might want to turn blue is to absorb more light and more heat. So like if mushrooms are about to freeze and they start to get frost, then that will break the cells and cause the blue staining, which will turn blue and absorb more heat from the light and, heat the mushroom up a little bit. So that is possible that it's for the blue color. Another idea is that these polymers will piss off insects that are trying to feed on the mushrooms, although that doesn't make a whole lot of sense because mushrooms benefit from getting eaten by insects and animals because they spread the spores around a lot more effectively than the wind. So there's a whole lot of different theories here, but it's definitely super interesting.
Yeah, it is super interesting. And I've heard some people suggest that these so let me check my term with you. Do you like this term secondary metabolites for referring to these other biochemicals?
Yeah, I think it's used pretty commonly all throughout biology.
All right, good. So these secondary metabolites, I've read some suggestion that perhaps they and also psilocybin increase under stress. Do you think that is true for both psilocybin and the other novel secondaries? And are there any insights to be adopted by home cultivators in this understanding?
I think it's true for cactus that when cacti get pissed off, they make more mescaline. And that makes a lot of sense because a lot of these alkaloids kind of like defend against predators and things like that. But I don't know that it's really true for mushrooms. I think if you anger a mushroom somehow, maybe you dry it out or bruise it or something, they don't really make more psilocybin in response to that. So I think it's more of a plant thing. And that makes sense just because, the mushrooms are not trying to fend off predators.
Mm-hmm, mm-hmm. Predators are mostly their friends because they're actually helping. Yeah. So similarly, I'd like to get your thoughts on the wild versus cultivated mushroom debate. We've already established that the metabolites secondary to psilocybin are important. Do you find that the wild versions of these mushrooms have more potency and variety than cultivated mushrooms that maybe they're not engaging with the environment in the same way?
Typically, yes, but probably not because they're wild. It's just that when you cultivate, you're always growing the same species 99% of the time. And so that's kind of limits the possibilities there. Whereas in the wild, you have so many different species that you're collecting. So there's a lot more chance to get different molecules. But also, Psilocybe cubensis is not a particularly potent mushroom. At least most of the strains, you get about, on an average cube, about 0.6% alkaloids by weight. Whereas, some of these other mushrooms, like the wild ones that people find often have 1% to 2% or even more alkaloids. So the wild ones tend to be stronger, but there's also very weak wild ones. Some of the Paneolus are very weak, and you would have to eat a whole lot of them to really figure out anything or really feel anything. But you don't really notice different effects from the same species growing indoors or outdoors. But that's so hard to quantify because even the exact same batch of mushrooms, if you were to powder it, homogenize it, and take it on different days, you would notice very different effects just because of all of, they're interacting with your body, they're interacting with all the chemicals in your brain, and maybe a whole lot of stuff in your subconscious has a big effect on what you experience. So, when you have even the same batch having such different effects on different days, it makes it really hard to quantify what the differences are. And, what really needs to be done is people need to try all of the alkaloids from fungi separately and in different combinations. And then to really score that, you would kind of have to have like a double-blind mushroom party where you had hundreds of people all getting different kinds of mushrooms, and then at the end you could give them a survey where they score a whole bunch of things from 1 to 10, and then you can crunch the numbers and maybe figure out something about the different effects of the different molecules. But I don't think anyone's ever done this on a large enough scale to make it statistically significant.
It sounds like it would be a very popular study, though. Would be no problem getting volunteers.
Yeah, I think it's the kind of thing you could do in somewhere like Colorado.
All right. So during the break, Alan, you and I were talking about these secondary metabolites and their value in medicine to people. And we started doing some of the comparisons between the cannabis plant and the mushroom medicine. And you started talking about the potential for mushroom medicine seems like it would be even greater than cannabis because more people you were suggesting might be able to get benefit from mushrooms. I don't want to put the words in your mouth, but you clearly have thought about this a lot. Would you go back into that for everybody? Because where you're going with it was very interesting.
Yeah, so everyone's heard of medical cannabis, and cannabis definitely does help some people. But I think most people who use cannabis just use it for the effects and don't really need the medicinal benefit from it. Whereas with mushrooms, I think the medicinal effects are very different, and they tend to treat things that a whole lot more people have. And so I think everybody has at least some sort of depression or addiction in their life. And those are two things that mushrooms really help a lot with. And it's kind of interesting how it works because depression is not well understood by scientists, but it seems that the psilocybin turns off part of the brain that causes depression. So people might feel some depression and then they will take some psilocybin and they don't feel, they feel all very trippy and they feel very different for a few hours, but then they wake up the next day and the depression is just sort of lifted. And it's an effect that usually lasts for months, sometimes years. And that's a whole lot better than a pharmaceutical antidepressant, because with these pharmaceuticals, they have all sorts of side effects and you have to take the pill every day. And if you stop taking it abruptly, you can have, really bad depression come back. Whereas, with mushrooms, just taking them a few times a year will often take care of people's depression in a much more natural and much healthier way than a lot of those SSRI medicines.
And certainly without a whole lot less side effects as well, because those SSRIs, if you can even convince somebody that they work, most of them have got pretty life dysfunctioning side effects, which, microdosing, that's very uncommon.
Yeah, exactly. And it's sort of the opposite with mushrooms, and that, people take mushrooms and they feel like they're more into life. They're more interested in connecting with other people. I find sometimes in my own life, I'll get kind of into a rut where I'm just sort of wasting my time and not really making really good use of my time. And if I take mushrooms, then I will wake up the next day and I will just be like the light seems a little brighter and I have more energy, and I'll just start working on stuff that I should have done. And the stuff that I'm working on, it feels really fun to work on, and I'll stay in that mindset for weeks, months. And, that's something super valuable. And, one of the worst things we can do as humans is to waste our time and not make very good use of our time. And the mushrooms really seem to help that quite a bit.
Here, here. One of the things that I vibe out of the mushroom scene that I haven't felt in a while is it feels a lot like some of the earlier days of cannabis medicine. As cannabis has become more normalized and more commoditized, a lot of the medical people have left, and a lot of the heart has left, and it's just becoming a lot more like pushing widgets out. And as we were talking about earlier, there's still not really a lot of mushroom in mushroom research and even necessarily teaching about mushrooms. And it seems to be still to hold a lot of that heart, because the people who are in it are in it for more heart reasons than spreadsheet reasons. And, I understand that, I'm a little jaded and I'll own that. But I think that the mushroom scene really feels like it's still steeped in science and heart and not been commoditized yet.
Yeah, and I think so. And I think there's several reasons for that. One of them is just the pharmacological effects of the mushrooms themselves. They kind of make you more in tune with nature and with life and your purpose and all of that. And then the other thing is just the capitalism aspect. It's very hard to make money doing mushrooms. So even growing psilocybin mushrooms, they're not really that profitable. And so the people that are really into mushrooms are into it because they are just really interested in nature and mushrooms themselves. They have more pure motives, whereas a lot of people grow weed for the money.
Mm-hmm, mm-hmm. So I've got these next 4 questions. They're kind of non sequiturs. They didn't really fit anywhere else in the show, but they are things that I'm either asked all the time or I have come up on other shows and we didn't have answers. And so the first one is it's we know that psilocybin mushrooms. Oh, no, wrong one. Sorry. So the enigma mushroom is what I want to talk about. The enigma mushroom looks like ribbons, right? Or even kind of like the human brain when it's grown. And it's become really famous recently, but it doesn't produce spores at all. And so my question for you is, from a biological standpoint, like, what kind of mutation is happening here, and would it ever even survive in the wild? It kind of feels like a lab mutation that needs to be held up to survive with crutches, or else it wouldn't.
It would not do well in the wild, and it turns out in nature, this sort of mutation is extremely common.
Oh.
That's one of the most common things that happens when you get a mutation, is that it interferes with the reproduction. So with plants, the seeds are not viable, or with mushrooms, it might not make spores, or it might make spores that don't germinate. Mm-hmm. And in nature, those sorts of things just die out naturally. But, in the laboratory, we can keep stuff like Enigma going. And nobody really knows what kind of mutation it is. That would be a cool one to do a full genome sequence on and try to decode the DNA and see if you can figure out what it is that's damaged about the genome of that compared to a regular cube, and figure out, what's going on. I noticed that these sorts of mushrooms, the mycelium, looks very different under a microscope. So typically, cubensis mycelium, you'll see all the hyphae, and they'll be very regular, kind of boxy with septa at regular intervals. Whereas with Enigma, if I take some of that mycelium and throw it under a microscope, I see the regular mycelium, but then I also see this much wider, thicker mycelium that has all sorts of little vacuoles in it that look like little dots along the mycelium. Mm-hmm. And I haven't seen that in any other mycelium, and I look at quite a bit of it. So that has to be, something to do with what it's doing, but what it means, I have no idea.
Right on. Fair enough. Cool. Thank you. So next non sequitur. We've talked about wood lovers quite a bit on Shaping Fire, and episode 113 with Christian Kalin, we talked about making a wood lover plot on your own property, which is a lot of fun. And right at the end of the episode, Christian mentioned something called Wood Lover's Paradise, right as we were ending the episode, and we had kind of run out of time. But I've been looking into it ever since that episode, and I was curious to know what your thoughts are on this, because there's not a lot of people talking about this idea that taking a wood lover mushroom has the side effects of temporary paralysis. And of course, the discussion is whether or not this is actual paralysis coming from the mushroom, or whether or not it's just somebody being so damn high that they're not moving. Would you please let us know what's going on inside the mycology world about this? Are people talking about this? Is this a thing? Is this just rumor for Instagram? What are your thoughts?
Oh, yeah. Christian's super cool. I visit him sometimes when I head through the Pacific Northwest. But the wood lover's paralysis is really a mystery, and I do believe it's a real thing. And what happens is people take certain mushrooms, typically ones that grow on wood, but it's not all of the ones that grow on wood. It's just a certain clade. And they will start to feel really strange, and then they won't be able to move. And this paralysis tends to stay around for quite a while after the psilocybin wears off. So I don't think it's an effect of the psilocybin, because if it was, it would wear off after 6 hours, and you would see it in cubensis. You know, people eat huge amount of cubensis all the time, and they never get paralyzed from it. You know, sometimes they get so high that they can't move, but once it wears off, you know, they're totally fine again. So the wood lover's paralysis happens with psilocybe azurescens, psilocybe cyanescens, psilocybe elenii, and there's one in New Zealand, psilocybe aurora, which causes it even more reliably than any of these other species. And they're all very closely related species.
Do we have conjecture on what the cause is, or is it we know it happens, but we don't have an explanation yet?
There's no shortage of conjecture on it. People that have run it through analytical chemistry equipment notice that there are some extra peaks on the graph that they get when they analyze a wood lover that they don't see with something like a Liberty Kappa or a cubensis. But really, you know, if you go on the internet, there's no shortage of people that tell you that they know exactly why, what causes wood lover's paralysis, but they all say something different. So, you know, I found that you can always tell that somebody doesn't know what they're talking about because they claim to know what causes wood lover's paralysis. You know, it might be a tryptamine, it might be some molecule that we just don't have any idea about. But it's certainly very mysterious and elusive, and making it harder to study. It doesn't happen every time. And sometimes two people will take a similar dose of the same mushrooms. One person will get it and one person will not. So I'm convinced it's a real thing, but whether we'll actually figure out what's behind it in our lifetimes, I am not so sure.
Right on. Great answer. All right. Next non sequitur. This is one that I'm asked all the time, and it's a very common debate in mushrooms, and that it is this. When we are taking our mushroom supplements, is it okay and appropriate to take one that is based just on mycelium, or do we really want to make sure that we're taking the fruiting body of the supplement?
Yeah, that's a really good question, and there's a lot of debates about that. And it's complicated because it's hard to say whether the mushroom supplements really do anything at all. And they definitely help some people sometimes. There's no doubt about that. But it's less clear which people should be taking mushroom supplements and for which conditions and how often they should take it and how much should they take. So, you know, with any medicine, what really matters is, did you get enough of the medicinal molecule into your bloodstream to have the effect that you're looking for? And everyone responds so much differently to all of these molecules. And in a mushroom, you're going to have hundreds of different molecules there, and they're present in different amounts with different growing conditions, different strains, and then different amounts of these molecules are present in the fruiting body and in the mycelium, and even different molecules in many cases. So a lot of times with these medicinal mushrooms, we don't really know which molecule it is that you actually need. And you might say like beta glucans, and that's a reasonable answer, but there's thousands of molecules that we call beta glucans. And then, like, how much of this molecule do you need, and which conditions does it cure? It's really hard to say. So, in general, it's going to be best to take both the fruiting body and the mycelium, and they don't seem to have a lot of negative side effects. So, more is generally better. I don't typically notice much effects. I think, one medicinal mushroom that definitely works are psilocybin mushrooms. I notice a lot of effects from those. And then the one that I think probably works is hericium. So the lion's mane, that has a nerve growth factor in it. And so that probably works, but there's a bunch of different nerve growth factors in the hericium mushroom that are present in different amounts and different strains. And so some that are in the mycelium and some in the fruiting body. So probably best to take some of both. And then there's other ones like turkey tail and Ganoderma and agarikon, and those definitely help some people sometimes, but it's much more nebulous exactly what they do. In animal studies, you can see that they're definitely antiviral, antibacterial, and stimulate the immune system and stuff like that. But something that's antiviral doesn't mean that you can take it if you have a virus and that you'll be better. And something that stimulates your immune system isn't always good for you to take. These mushrooms typically are healthy, and people that take them are, generally don't have bad effects of them. But exactly what they do and who should take them and how much of each molecule they need is just almost impossible to know. And, there's thousands of diseases that are known. So to even test one mushroom against thousands of diseases would be thousands of clinical trials. So you're talking $1 trillion right there. And then to test, every chemical in each mushroom against every disease, that's just like a huge amount of testing. And then to test every mushroom against every disease would cost more than all of the money that's ever been printed in the history of humanity. So we kind of have to look to nature to get clues as to which mushrooms might be useful for which things, or look to the native cultures who have been using them for thousands of years and have figured some of this out. But for the most part, it's not figured out, and it's definitely not figured out to the point where anybody can say that mycelium or fruit body is better, but definitely can't hurt to take both.
Damn, that's the best, most comprehensive answer that I have gotten. Thank you for that, Alan. So I want to shine some light on what kind of feels like a side quest of yours for other people. But from talking to you, I know that it's very much at the central of what you care about, and that's your Python programming. You have put out a whole bunch of software tools for mycologists, and I want to make sure that we take a moment and plug those for anybody who's listening, so they can take advantage of this important work that you're doing. I'm not even exactly sure how to ask you the question other than to say, will you tell us about the tools that you've released?
Yeah, and so, I've been programming stuff since I was like 5 years old, but I didn't get into it really heavily until more recently. All these AI tools make it so instead of taking a month to code something, I can do it in just a couple minutes. And so I've written a bunch of stuff that I think is useful for people, and just like whenever I need a program and there isn't a program to do it, I'll just write it myself. And I encourage other people to experiment with this too. Even if you don't know any programming, you can still use these tools to make really useful programs. But for someone who does know programming, then these tools are even more useful because they just cut the amount of time that it takes to do these things. So probably the coolest thing that I've written is a replacement for Adobe Lightroom. And so I use Photoshop and Lightroom for my editing, and I have been, Lightroom has not really been perfect for what I want to do. And, what I do is just take a camera out into the woods and try to get the best picture I can of everything that I see in nature. And then when I get back home, I want to get all of those photos uploaded to iNaturalist, Mushroom Observer, Instagram, LinkedIn, all of the other places that people upload their photos. And so you can do it with Lightroom, but it's kind of clunky. And so I decided to make my own. And so I've called this Fast Stack. And fast because I wanted to make it so I can flip through the photos very quickly, and I do a lot of photo stacking. So this one is available for GitHub. Really everything I write is, I just release it for free under the MIT license, which is the freest license there is. So anybody can use the software for free. Anybody can edit it. People can even take my software and sell it. They don't have to give me money or anything like that. It's just completely free software. But Fast Stack, I've been working on it for about 6 months now, and it has come along. So it's actually really useful. So kind of does the same thing as Lightroom in that it shows you your photos. But one thing that's much better is that you can drag and drop directly into a web browser. So I can just mark a whole bunch of my photos and drag and drop them into iNaturalist or Mushroom Observer and upload the photos there. Or I can mark a bunch of them and drag them into Instagram to make an Instagram post. And then also it keeps track of how far I've gone in each directory. So each folder is a different day's worth of photos. And when I look at all my folders, it shows me exactly how far I've gotten, so I can really easily see like how much I've uploaded in each directory.
Right on. I believe that you have another piece of software that you put out that is for DNA barcoding too, right?
Yeah, so the DNA barcoding one is a website. And so I wrote it in Python, but instead of just distributing the Python code, I made a website and have that run in the back end. And that is called Dicaria. So it's dicaria.us. And this one is for building phylogenetic trees. And so there's already a couple websites like this, but they're really clunky and kind of hard to use. And so this one, you can give it an iNaturalist URL or a MycoMap URL, and it'll build a phylogenetic tree out of the DNA sequences on that URL. So you can see exactly how the different things are related. And you can use it for anything. You can use it for plants, viruses, bacteria. I use it for fungi, but it makes it so, it used to be to make a phylogenetic tree, you had to edit all these text files and then run all these command line tools. And it was very time consuming just to edit the files and then make the tree and then change something and make it again. Mm-hmm. And so now it's very quick. It's very quick to edit them. Anybody can do it just on their phone. You don't have to know anything about DNA to make a good tree. And so, that one is still in active development, but it's to the point that I use it all the time. And if I just want to answer a question like, well, how many Russula are there in California and how are they related? I'll just search for Russula on iNaturalist and just pull up the observations with DNA barcodes and then paste that URL into the Dicaria website, and it'll download all those sequences from iNaturalist and calculate how they're all related and then build a graphical tree showing exactly how many species we have. And it's so much better than trying to get that information from a field guide, because the field guides will be all out of date. This has all of the very up to date, all of the undescribed species that are not in any of the books are all in there. And so that works pretty well, and it just makes it really easy for people to build phylogenetic trees. And then the one thing that people use more than anything else is probably the iNaturalist herbarium label generator. And so that one, I have it on It's up on images.mushroomobserver.org/labels, but you can find them at GitHub as well. But it's a website that you enter an iNaturalist or Mushroom Observer observation number, and then it goes out to iNaturalist or Mushroom Observer, downloads a whole bunch of data about your observation, and then prints out a really professional looking label with a QR code that you can print out. And so I have thousands of bags of mushrooms here for study, and I'll make a label for each one. And then, they look really nice. I print them on cotton cardstock, so the label will last for thousands of years in an herbarium. And when I send it in for DNA sequencing, they don't have to type in the 9-digit number for every one of my collections. They can just scan it real quick with the barcode scanner and check them in really quickly. And so that's one that a lot of different people use to organize the herbarium and make labels. And then I also have the source code available, so you can download that and run it locally if you want.
Right on. That sounds like a great useful tool, and especially having the ability to interact with the more active, live, living database instead of, trying to deal with paper books or even outmoded, websites or forums. I also want to talk about ultraviolet flashlights and why they are useful. I finally got one just a few months ago, and I picked up a cheapie for $16, and I'm like, oh, this is really cool. I definitely need a much nicer one. And so then I upgraded, and I see that the ones that I upgraded to, you have back on your link tree as well. So you've got them back in stock. So I wanted to plug them because there really is such a difference between using a UV flashlight that's just got some cheap LEDs in it versus using a higher end one that has got the professional, maybe even patented filter on the front. And, at 365 nm, it really makes everything in the forest pop. But since I'm not as hardcore of a mycologist as you, I don't really know how to describe to people like the best uses for it as far as hunting for and researching mushrooms. So will you just kind of talk about the usability of these ultraviolet flashlights? Because I can talk about how fun they are and how badass they are in tide pools as well, because all of the everything with a exoskeleton just pops up, but I'm not as experienced with mushrooms. So will you pick that up?
Yeah, so it turns out that in nature, there's a ton of fluorescent molecules. And a fluorescent molecule is just a chemical where when ultraviolet light hits it, it re-emits a longer wavelength photon that's in the visible spectrum. So sort of like a chemical sensor in that you can shine it on different things, and some things don't light up at all, whereas other things just light up like crazy. And that's due to the presence of fluorescent chemicals. And they are really cool for making artwork in that you can like shine it on things and get these otherworldly colors. But they're also really good for science in that you can detect the presence of these chemicals that normally you just wouldn't be able to notice that they're there. And so sometimes I'll be in the woods and I'll find a mushroom and I won't know quite what it is, and I'll shine the UV light on it, and I'll be like, oh yeah, it's one of those, and I'll just instantly know what it is based on the color that it lights up. And so I do a whole bunch of ultraviolet photography, and it's really easy to get a good ultraviolet photo from a cell phone or from a professional camera setup. And what you see really depends a lot on where you are. Like if you're out in the desert, everything out in the desert has a lot of ultraviolet protection, so none of the plants light up at all, and instead you'll see certain minerals lighting up, and a lot of insects. The scorpions light up like crazy. Whereas if you go deep in the forest, then those plants don't need the ultraviolet protection. So you see a whole lot more fluorescence, a lot more lights up. You get completely different colors off of the different plants and lichens. Some of the flowers look spectacular because they have fluorescent pollen. And then if you go into the Amazon rainforest, shining it around is almost like fireworks. Almost everything lights up and you get every every color in the rainbow. Just everything is covered in something that's fluorescent there.
Oh man, the rainforest. That sounds really, really cool to get all of that. I like your description of fireworks. While I have not been to the rainforest with it, I mean, except for, I guess I live in a rainforest, but I haven't been over to the Ho with it yet. But here on the island, I've taken it into the forests, and it's amazing when mushrooms suddenly are glowing from, 10, 12 feet away. It's almost like you shine the light and then it like waves to you. It's like, oh, I'm over here, because they really stand out. And what really stands out here also are all of our ferns. The bottom side of ferns are all red. So you see all these red plants, which is really extraordinary. So yeah, I recommend it to folks, like, just if you like nature, full stop, right? And if you also want to go into mushrooms or tide pools, or it's also good to have it, but I recommend everyone gets one. And the Conroy ones that you sell on your LinkedIn are fabulous because, again, they've got that specialty filter. They're not just made with cheap LEDs.
Yeah, so if you're going to get a UV light, the most important thing is the wavelength. So if you go to a party, they will have these purple ultraviolet lights, and that's 395 nanometers. And those are very cheap, but if you take them out into the woods, they're pretty disappointing.
Yeah.
The other wavelength that's common is 365, and a 365 light looks a whole lot more blue. It looks a whole lot less purple, and it's further outside of what your eye can see. So when you're looking at something, you're really only seeing the fluorescence instead of seeing the light from your light. So you definitely want to get a 365 light and not a 395 light. And then also just like a really bright one. So there's a lot of really good ones on Amazon, or I sell really nice ones that are branded with my company, Mycena. It doesn't really matter where you get it, but just a really bright 365 nanometer light makes it really cool to be in the woods at night.
Excellent. Thank you. So, Alan, let's finish off with this. So many people are looking to reconnect with themselves and with the earth and with nature. And, some people are doing it because they're bored with their life. Other people because of the life stresses. There's just a lot of good reasons to go into nature. When you are talking with somebody and you're warmly encouraging them to lean into mushrooms, I'd like to hear what you say to people. Let's end this with a call to arms for mushrooms to get out and do some foraging. What do you tell people to kind of like encourage them to, get out from behind their computer and get out there and look at mushrooms?
Yeah, I think there's something in our DNA that makes us really enjoy being in nature. And, it's partly because we evolved in nature, and partly just because everything in nature gets more and more interesting the closer you look at it and the better you know it. And it's sort of like a never-ending fractal, where the closer you look, the more detail you see, and that never really ends, even with microscopes and even beyond. There's just so much to see out there. And it's also just really good for your mental health to get outside and spend a lot of time in nature. And that's one of the reasons I quit my job like 15 years ago and decided to be a mycologist full time, is because I just wanted to spend more time outside in cool places. So the main thing that I think people should do is use the iNaturalist app. So iNaturalist is an app and it's a website, and the point of iNaturalist is to bring people closer to nature, and it does a really good job of that. So the idea is that you can take a picture of anything in nature and add it to iNaturalist, and then it will try to automatically identify it using computer vision. And so if you have a really good photo of a mushroom, it'll be correct, 95% of the time. But the really cool thing about it is that it makes a permanent record. And so these records are publicly accessible, and people will come along and identify your mushrooms for you. Or anytime I see a plant and I don't know what the plant is, I'll put it on iNaturalist, and people will come along, and the botanist will tell me exactly what plant I have. So with mushrooms, it's really important to get one photo that has the underside and the top of the mushroom in the same photo. With plants, you want one photo that has the leaf and the flower in the same photo. But, anything is really cool to add to that. And if people have a bunch of stuff on their iNaturalist, they can message me their username, and I'll flip through their mushrooms and try to put names on stuff.
Right on. Cool. Thank you, Alan. And thank you, Alan, for spending your time with us here on Shaping Fire today. I know that you are a very busy cat traveling around, and your time is very valuable. And we all appreciate that you peeled off some time for us today to, share your stories and your insights, and actually probably just get most of us really jazzed about mushrooms. So thank you very much.
Thanks for having me on.
Excellent. So friends, if you want to follow along with Alan Rockefeller, I highly recommend that you follow his Instagram. It is one of my favorites, and that's at Alan, ALAN _ Rockefeller, ROCKEFELLER, Alan _ Rockefeller on IG. From his LinkedIn on his Instagram, you can find everything else you want. You can find the ultraviolet flashlight we were talking about, and his DNA barcode coding at home stuff, links to his YouTube videos, which are also great, as well as his iNaturalist profile, and links to his website at mycena.llc, where you can book an upcoming trip to Costa Rica with him and others to do some mushrooming. So that is all at Alan _ Rockefeller on his IG. So thank you for being with us. This has been Shaping Fire, episode 128. You can find more episodes of the Shaping Fire podcast and subscribe to the show at shapingfire.com and wherever you get your podcasts. If you enjoyed the show, we'd really appreciate it if you would leave a positive review of the podcast wherever you download. Your review will help others find the show so they can enjoy it too. On the Shaping Fire website, you can also subscribe to the newsletter for insights into the latest cannabis news, exclusive videos, and giveaways. On the Shaping Fire website, you'll also find transcripts of today's podcast as well. Be sure to follow on Instagram for all original content not found on the podcast. That's at Shaping Fire and at Shango Los on Instagram. Be sure to check out the Shaping Fire YouTube channel for exclusive interviews, farm tours, and cannabis lectures. Does your company want to reach our national audience of cannabis enthusiasts? Email hotspot@shapingfire.com to find out how. Thanks for listening to Shaping Fire. I've been your host, Shango Los.